Hi, welcome to Swift Biosciences webinar on sequencing SARS-CoV-2 and identifying variants of concern. For those of you who may not know, Swift was recently acquired by IDT. We're very excited about this transition where we can offer a wide variety of support and competitive pricing for our customers. My name is Jordan RoseFigura, and I'm the Director of Material Sciences at Swift Biosciences. Swift offers a wide range of products for NGS applications, from genome sequencing to epigenomic sequencing, RNA sequencing, and library normalization. Today, I'll be talking about our targeted sequencing, specifically our XCell Amplicon panels and our Swift Normalase Amplicon panels, also known as our SNAP panels. The last year has been hard on everyone with the coronavirus pandemic, and surveillance of SARS-CoV-2 and finding new variants and tracking lineages is of supreme importance in our new world. The Swift Normalase Amplicon panels, SNAP panels, are an efficient and easy way to do this. The SNAP workflow consists of going from DNA to a sequence-ready library. It involves a multiplex PCR step, followed by adapter attachment and indexing PCR, at which point you have sequenceable libraries. After that, and included in the kit, are the reagents for Normalase, which is optional, and I'll be discussing that more later. The SARS-CoV-2 genome is an RNA genome of about 30 kilobases in length. The Swift SARS-CoV-2 Extended Genomic Coverage panel uses this roughly 2-hour SNAP workflow and covers 99.7% of the SARS-CoV-2 genome. This is done with 345 overlapping amplicons that have an average size of 150 base pairs. As an input material, it uses first or second strand cDNA, and we validated down to 10 viral copies. Swift has available 384 CDIs, 384 UDIs, and we're looking to increase that UDI multiplexing capacity very soon. Our kit is flexible for reverse transcriptase kits. Most people just make the first strand cDNA and go straight into our multiplex PCR. There's a wide variety of compatible RT modules as long as they're using random primers and has a processivity greater than a kilobase. Here I'm showing an IGV shot of the results from our panel covering the full SARS-CoV-2 genome. In spring of 2020, so last year, we initially launched a version 1 of this panel. And then in the late summer of last year, we launched our extended genomic coverage panel, which filled in a few gaps in coverage and extended the coverage on the 5' and 3' ends. The data I'm showing here is from a nasopharyngeal swab, where the library was generated with the SNAP SARS kit and sequenced on an aluminum-iSeq. Our assay is very flexible and supports a variety of applications and sample types. It can be used for viral surveillance, wastewater, epidemiology, variant calling, and sample types that it's been successfully used with include nasal swabs, stools, and sputa. We've had a lot of great partners we've worked with on the development of this panel, including Dr. Adriana Higuet at NYU, who was able to, early on in this pandemic, track a variant of interest. In this small study, she used 29 nasopharyngeal swab specimens and was able to find this A to G transit variant and use that to track the phylogeny of samples around the globe and see how this D614G allele spread. We have validated this kit across a wide variety of viral copy numbers. This is data made at Swift using a synthetic SARS-CoV-2 control in a background of human UHR, right? And we were able to get a very effective percent read alignment that didn't change with read depth. The data here is shown for 1 million reads in purple and 50,000 reads in blue. We were also able to get bases greater than 10x coverage at very high amounts. This does drop off at the lower viral titers. So one thing Swift did when we launched the version 2 of our panel was also develop and include cycling recommendations to optimize data at the lower viral titers. So, and what this data did, it's the same workflow, just changing the cycling conditions. And you can see that it very much increases the percent reads aligned and the coverage at the low viral titers. So the low input protocol is shown in purple, while our standard condition is shown in blue. We recommend this low input protocol for samples with less than about 1,000 viral copies. So, but here I'm showing data for 1 million viral copies and 1,000 viral copies using the low input protocol to show that we have very robust coverage over a range of viral titers. So this is an IGV shot of the whole SARS-CoV-2 genome with, zooming in on a range so you can see the coverage a little more clearly, right? We have very good coverage uniform and very good coverage with 1 million copies, and that is still robust at 1,000 copies. This data was generated using the BEI irradiated standard and sequenced on an Illumina mini-seq with 2x150 paradigm sequencing. As I mentioned earlier, the SARS-CoV-2 panel has an average of 150 base pair amplicons. This smaller amplicon size makes this assay very robust for a wide variety of samples, including damaged or degraded samples. There's been a couple papers recently using our kit with wastewater, including this paper from ASU that used our kit to determine lineages in 91 wastewater samples, and they were able to sequence samples and determine the lineage for a sample with CTs up to 35. So we were very excited by this. We think it's very important work and really shows the power of the SNAP SARS kit. Related to wastewater, we had some inquiries about whether we could make a panel that sequences just the S gene. This is a way to sequence the S gene and get really deep sequencing to look at subpopulations without spending a lot on your sequencing reagents. So the S gene panel covers 100% of the SARS-CoV-2 S gene with 43 amplicons. It is the same workflow as our full genome panel. It just requires less sequencing. So here's an IGV shot showing our comprehensive coverage of the SARS-CoV-2 S gene. We have 100% coverage of it. This data was generated with 1 million copies of the BEI standard. And here I'm showing a dot plot where each amplicon in the panel is represented by a blue dot. The blue line on the top is 5x the mean coverage, while the red line is 0.5x the mean coverage. And as you can see, we have a very tight coverage uniformity in this panel. Swift has also tried to support our customers by developing data analysis tools for our amplicon panels. We have a analysis pipeline available as an open source tool. It is Dockerized, and there is demo data available. We suggest you can go to the website shown here. Our data analysis tool performs primer trimming, alignment with BWA, followed by primer trimming with primer clip, which is a tool developed by Swift specific for our one-tube overlapping amplicons. The pipeline then generates on-target and coverage metrics, and then performs variant calling. We have a tech note for our SARS-CoV-2 Dockerized data analysis. I'm showing it here. And this is just another workflow to show what this pipeline does. One thing that we have added specific for SARS-CoV-2 is consensus calling. So our pipeline generates a consensus FASTA, and also gives a report on variant designation using Nextclade and Pangolin. Next, I wanted to talk a little bit about the Swift technology that allows a one-tube multiplex PCR and the benefits that can come with it. So if you have overlapping amplicons in a single tube, they can create a variety of species. I'm showing a cartoon here just showing two overlapping amplicons where the primers are shown as arrows on the top. Right, we have two sets, in the first one in blue and the second one in purple. These can create our what we call intended amplicons, shown on the bottom in blue and purple. They can also create a super amplicon where forward one in blue and reverse two in purple interact and form what is almost a double-sized amplicon. There's also what we call mini amplicons, where forward two and reverse one interact and form a short amplicon. Now, if you just took target-specific primers that made overlapping amplicons and threw them in a tube, that mini amplicon could very easily take over your data. Because of its size, it PCRs up much more efficiently. Part of Swift's multiplex technology is that we stop that from happening. So what I'm showing on the left is a cartoon which is representative of data you might get if you just threw these primers in a tube. On the right, I'm showing data, a cartoon of what's much more representative data from Swift's multiplex technology. You don't want these mini amplicons because they don't give you as much data. They can overcluster a flow cell, and it's just a waste of reads. Here I'm showing some actual data. This is from Swift's 56G panel, where I'm showing two locuses in PIC3CA. One where there are two amplicons that are overlapping. This is on the left. And as you can see, there we see the super amplicons on the top, the intended amplicons, and then just a couple of mini amplicons. But they definitely have not caused a problem in the data. On the right, we're showing amplicons that are neighboring, so they're not overlapping. And as you can see, we still see the super amplicons on the top and the intended amplicons, but no mini amplicons are formed. So just to back up again to this, now that we've dealt with the mini amplicon issue, to back up to what can overlapping primers bring to your data. So in either a one-tube or two-tube solution, Swift Biosciences has a one-tube solution where we form this super amplicon, you will get overlapping primers. In a two-tube solution, this super amplicon won't form. And that's because forward one and reverse two are in different tubes, right? So there's no super amplicon. And you may be thinking, Jordan, why do I care? Well, I believe that the super amplicon actually brings important data and is a nice feature to have, because what happens if a variant occurs under a primer binding site? There are new variants all the time for SARS-CoV-2, and it isn't viable for all labs to be versioning their primer set every time a new variant occurs. So what happens if a variant occurs under a primer binding site? I'm showing a variant as a red star here, and it's occurring under the forward 2 primer. So that's been grayed out. It's caused this, whatever this variant is, it's caused the loss in function. That means that that purple intended amplicon can't form, right? Because the forward primer is no longer functional. You will still see that variant in either a 1-tube or 2-tube solution because it will be covered by the neighboring intended amplicon, shown in blue here. But what happens to that coverage over on the right of this slide, that was covered by the purple amplicon initially? Well, if you have a 1-tube solution that creates a super amplicon, you will not only also see that variant in the super amplicon, you can attain coverage using that super amplicon and maintain full genome coverage. The UK variant is an excellent example of a case where this has happened. The deletion 1128.8 to 1129.6 occurred in our assay under a pre-existing primer site, and you can see that in the data that I'm showing here. This is IGV. The target regions are shown in the lighter blue on the bottom, and the primers are shown in dark blue. So you're seeing that that deletion happens right at the 3' end of this primer, the primer on the left. So, but in our data, we can see that deletion clearly from the targeted region, but we maintain coverage, even though that primer is not functional because its 3' end cannot bind. So how do we do that? With the super amplicon, so I've just zoomed out in this IGV shot of it. So you can see that deletion kind of right in the middle of the IGV shot, and you can see that there are super amplicons throughout the data, and that's where our coverage comes from. So we think that having this overlapping amplicon 1-tube solution is very powerful for discovery of new variants and adding robustness in the presence of variants. So just to change gears a little bit, I had promised that I would talk about Normalase, and I will do that now. The Normalase technology is specific to Swift, and it is viable for a lot of different assays. Most of these Swift kits have Normalase as an optional workflow. So in SNAP, the Normalase portion is, as I said, is optional and occurs after indexing PCR. So you have full libraries that are sequenceable, and you can normalize them if you choose to. The Normalase workflow is very straightforward. It involves a Normalase 1 step, equal volume pooling of all the libraries, so it really lends itself to high throughput, followed by Normalase 2 that occurs on that library pool, and then an inactivation. And after that, you can take this library pool, denature it, and stick it on your Illumina instrument. Right? Part of how this works is that in Normalase 1, it is a selection step. At that point, you then take and pool all your libraries. So showing here, it's all in one bar because all the libraries are together. And then in Normalase 2, only the molecules that were selected are represented in your final pool. This is a really great fast technology. It's easily automatable, and it really improves the read depth variation. so CV coefficient of variation within a pool, which allows for savings on cost of sequencing. So here I'm showing some data of showing an improved index balance between Normalase and qPCR. So what we did was we made Normalase libraries. All these libraries were above the required yield minimum, shown in a green line. And we pooled and sequenced them based on qPCR, that's shown on the right, where we got a CV of almost 24%. We then took the same libraries, normalized them, and sequenced them, and got a CV of slightly less than 6%. So as you can see, you get much more even coverage on a per library basis with Normalase chemistry. In addition, Normalase has the added benefit of mitigating index hopping on pattern flow cells. So the data I'm showing here is from an iSeq, and you can see that libraries were all on a per read basis, equally identified, but that without Normalase, the index hopping was present, shown as the purple dots. But when you add Normalase, that's on the left, index hopping drops down to almost unmeasurable levels. So that's just really an added benefit to Swift Normalase chemistry. Next, I wanted to talk about resolving S gene dropouts in COVID-19 samples. So to look into this, Swift worked with gene markers that's based in Lansing, Michigan, and identified 8 samples from Kalamazoo. These samples were S gene target failures. Right in their PCR assay, it's known that the TaqPath assay can have S gene dropouts with the UK variant. So we collaborated with gene markers to identify the lineage of each of these samples. We used both the SNAP SARS-CoV-2 and the S gene panels just to try to see what depth do we need, looking at real samples. We assumed that all these S gene dropouts were the UK variant, B117, right? But what we found was that sample 8 was actually an unexpected variant. So I'm going to discuss that a little more. But I think this really shows the importance of sequencing, because working on the assumption that all the S gene dropouts were the UK variant would have resulted in misreporting for this variant. And that can complicate disease tracking and all sorts of things. So looking at the data, we're able to see that, so what I'm showing here is 2 of our other samples of the first 2 IGV tracks, and then sample 8 as the 3rd IGV track. And you can clearly see that deletion 69 to 70 on the left is present in sample 8, but deletion Y144 is not observed in sample 8. And that was one of our first indications that this sample was not the UK variant. This variant again, or sample 8 again, didn't have N501Y, which we know is an important mutation. There was enough depth at that locus in sample 8 for appropriate germline variant calling, but it was all wild type. We did not see the N501Y, which we saw in the other UK variant samples. In addition, there were other mutations observed in sample 8 that were not known UK variants and were not seen in the other samples. So what was this? Sample 8 ended up being B1375, which is not a variant of concern, right? So, and I'm showing here a phylogeny map from NextClade, and you can see that red dot is sample 8, right? And it, and I think this just again brings home that this could have been misreported based on the PCR results alone, but that sequencing really made it clear what this sample was and what it wasn't. So Swift, just to wrap up, Swift is really focused on empowering data, right, by sequencing. We also, in addition to our SNAP SARS assay, we also have RNA kits. If someone's looking to do RNA-seq, we can make libraries from as little as 10 nanograms of total RNA. Our Swift Excel NGS methyl-seq product can unlock epigenetic insights, so for methylation of a sample. And we can also do host research with generating whole genome libraries from as little as 1 nanogram with our Swift Sonic 2S library prep. Thank you for listening to this webinar. I hope you learned something, and please get in contact if you have any questions or would like to learn more. Thank you.